few logistics. We ask you to reserve your questions to the end, and you know, good questions. And we'll have a seminar and questions, and then after that, the committee will we will ask the committee to hold out your questions to the best part, and then other people ask questions here. And we know from past experience, she's good questions. So on that note, I'll let Dr. Tierney, her advisor, introduce.
Yeah, so thank you all for coming. For those of you who are here, I don't know how many are online, but it's great to see people basically as well. And even if it means that trying to plan here, I think take too long. We've got an hour. But I was thinking maybe I should use we call it all our accomplishment. I don't know how to use Nicole, I met Nicole when she was in first year of the BBM program. Still in the back, she was knitting, she was speaking infectious diseases. And then you walk by her and she'll say, "What about microglia?" At that point, she was interested in microglia. She was interested in inflammation of the brain. And then you look at her resume, yeah, she's been doing some of this before. And then one fine day, she came to my lab and said, "I want to grow microglia." And he told me, "Why do you want to grow microglia?" "Well, I want to see if it has anything to do with addiction." Oh, you showed her how to grow microglia. Eventually, she came back and said, "I want to work on it. And I want to work on addiction." I'm like, "We don't do addiction. We don't do anything." And usually, when graduate students come in and ask you for treatment, they tell you something that's all comfortable with. Yeah, you would take that from them. So we got into addiction, and Paul's going to talk to you about what she's been doing for gruesome years. I don't know if it's gruesome because she was working with a authority. Gruesome because COVID addiction, couldn't do a lot of the work. Or just that it just hates. So I'm really excited to introduce her and tell you I will tell you about it. She's done some amazing work. All that into doing addiction work. But most importantly, I think this is one of the first times where, one of the few times, that we're actually connecting inflammatory responses and behavioral responses. Not really drawing a full line, but at least starting to figure out. I will without saying too much more, I'll let her talk about this. And thank you very much for coming.
All right, thanks, Max. I do want to mention to the people online, my husband is monitoring the chat and will relay your questions if you have any questions to write down. Also, for the people who are not going to be in here for the defense portion afterwards, in the niche, we will have snacks and drinks and such for you guys to hang out. So feel free to hang out in there and chill with my parents and such. So I am super excited to share with you some of the highlights from my work for the past 4 years at this point, entitled Injury-Induced Neuroinflammation Alters Addiction Behavior. If you do want to learn anything more about this or go into more detail about our work, we do have some publications coming out early next year. I will warn you, there's a lot of supplemental figures, and if you ask Max, he's going to quiz you on the supplemental figures before he answers any of your questions about the work. So, all right. So let's jump right in. What I'm going to talk to you about today is, first of all, why these problems are significant, why they're important to our lab, and then I'm going to highlight our research studies. So 3 of our main studies that we've done in the lab to date. Then I'll talk about some conclusions, and so what exactly my project amounts to for our lab and going forward for some of these studies here. So first, our significance. traumatic brain injury is a worldwide problem, also a national problem, with 64 to 74 million people affected every year and 5 million in the US annually. But these numbers are based on the diagnoses, the burden on the hospitals, and then that cost ratio for medical departments. And so this could be vastly underestimated, because mild traumatic brain injury accounts for 75 to 90% of your injuries, and they are the hardest for not only doctors to diagnose, but even patients to identify. I mean, last time you guys hit your head on the cabinet, you run to your doctor and go, oh my God, I hit my head. No, you probably took some aspirin if you got a headache, but you went on with your day. So you didn't tell anybody about it. And so really, we have a lot more people at risk. And when you get even a mild injury, you're going to have the risk of Alzheimer's, Parkinson's disease, PTSD, depression. And then for me, I'm really interested in addiction. And so substance use disorder that develops in our patients here. With a history of TBI, you actually have an increased risk of about 3 to 6 times out of the general population to develop a substance use disorder to any substance that's defined here. However, it becomes really important when we start talking about the opiate epidemic. So we know the numbers of that is rising in the nation, and it's a big burden on our system. Our adolescents and our soldiers are at really high risk, partially because they play sports. They're deployed to war zones where they're getting constant head injuries. So when we look at that population, we actually see that adolescents with TBI have a risk of 2 times or more of opiate use disorder, and then soldiers have a risk of 3 times or more. And so that's really significant within these populations. So let's talk a little bit about what happens to your brain when you get a traumatic brain injury. So first, you're going to get your primary injury. You're going to hit your head. You're going to get some physical damage to your brain. So that could be cellular damage. You could get some axonal shearing, some damage to those cells in your brain. You can also get some blood-brain barrier disruption. You can also get tissue deformation. And so all of these things are going to happen. There's going to be release factors. There's going to be some debris in that area, right? So then your secondary injury comes along. So the secondary injury is really when that immune response comes in. You might get some ischemia in the area, so lack of oxygen to that area, and you're going to get an increase in the damage site. So that damage is actually going to spread from the main injury site, and that's partially due to the immune response. That's partially due to some of the factors that are released during your primary injury. So some of our cells that are really important there are going to be our macrophages and microglia. And so we love macrophages and microglia in the Sheeran lab. They're our favorite cell types over there. So what they do is they remove the cellular debris. They can repair damage. They can increase synaptic pruning. And so what that actually means is those neurons in your brain that really control a lot of what you do, how you think, how you process, those cells can actually be physically altered by your macrophages and microglia. Then you also have the production of cytokines that can happen as well. And so those can actually have direct effects on those cells, like trafficking of AMPA receptors to those synapses, changing the way that neurons actually interact with each other. And so changing your processing that can happen there. So that's going to be some of the things that you're going to see with this cellular population. That's one of our ones that is going to come in to all of our studies and really be a main feature of this presentation. When we look at our response to TBI, you are going to get structural alterations that happen actually at the site, right? That makes sense because we have some physical damage that's happening to your brain. You're going to have some reorganization. You're going to have some changes that happen there. However, you're going to get some changes to deeper brain structures. And so that's really what we we're interested in. We want to understand how that happens, and we want to know, understand where that immune response is actually directed to. When we look at human studies, you're going to have some differences that happens in your thalamus in advanced MRI studies that are happening. And so your thalamus is really going to be your relay center for a lot of the different systems. As you can see on this, you have a lot of different systems that go through your thalamus here. And then also you can have some changes to your ventral midbrain, and that's a lot of where your reward pathway is. So your reward pathway, very important to the development of substance use disorder. So we're going to come back to some of these structures. And the VTA, or ventral tegmental area, is going to be really important to opiates, because that's really where it's going to assert a lot of that action that we're going to have. It's going to talk to your nucleus accumbens, where you have that main reward pathway center that we see changes in with any type of substance use. So from here, we identified that there are some gaps that exist within the research. What we understand is that there is inflammation-induced changes within the limbic area of the brain that are essential to the development of drug-seeking behavior and escalation. We know this from the studies that involve ethanol or alcohol use disorder. And then we also know this from cocaine studies. And so we've seen that modulation of the immune response actually changes drug-seeking behavior in these, in our test subjects, mainly rats and mice are what you're going to use for these areas. And so what we want to understand is opiate use disorder, because alcohol interacts with the brain very differently from a lot of our other substances. And then cocaine is a stimulant, and so it acts in a really different way than our opiates do. So we want to understand how does the inflammatory response relate to changes seen post TBI at sites distal to that impact site. So you get a hit on your cortical area. How does that translate to differences that you're seeing in your deeper brain structures, even months after your injury, and no matter where you actually get hit on your head. We also want to understand what is the impact of these changes on the increased risk for opiate substance use disorder. So our main research question is going to be, are the rewarding effects of opiates enhanced by the inflammatory response to mild TBI? So that brings us to our first research study. It's entitled Neuroinflammatory Response to Mild TBI is Associated with Long-Term Behavioral Deficits. Now, this is really a model development paper that our lab first participated in. So my first challenge was trying to develop a model that had a mild injury, had long-term deficits that we see, and then long-term chronic inflammation. And so that was really the goal of this here. We decided to go with mice for our study. And so you'll be seeing some mice data. So our hypothesis is that the acute pro-inflammatory response to mild TBI will extend distally from the site of injury to affect deeper brain structures, resulting in chronic inflammation associated with long-term behavioral outcomes. So this is the model that we developed to go ahead and understand this. So first, our animals arrive. We do some baseline testing and some training, so that we can actually have our animals perform different assays for us, so we can understand deficits in both motor and cognitive function. Then we go ahead and induce our TBI injury. And so that's going to be discussed in just a second. Then at our 6 and 24-hour time point, we start to interrogate our inflammatory response there. At 2 days post-injury, we start doing our behavioral testing, and then we continue both of those all the way out to day 30. So all the way out to that chronic response. And with mice, they live a little bit less time. And so 30 days really is that chronic response for us that we can see here. So when we were discussing what type of injury we wanted to do, I wanted a really reproducible injury that was really consistent within our animals. And so I chose open controlled cortical impact model. So what this does is we actually, you can see my little mouse, he's in a stereotaxic frame. So he's anesthetized, his head is held in place, and then we drill a craniotomy in his skull. That piston that is featured right here, that we can control the depth that it goes down into the brain. We can control the dwell, so how long it's in the brain. We can also control the velocity, so we can make different types of injury parameters. And so I studied mild. We also have moderate and severe models that are present within our lab. Our sham animals, or our control animals, actually receive a craniotomy so that we can control for any type of inflammation caused by drilling a hole into the brain, into the skull. But they don't receive any type of impact there. So they still have that surgical procedure on board. Now, when we're looking at what actually happens to the brain when we injure it, we injured on the right side, we centered over the primary and secondary motor cortices. Now, this was really important to us because we wanted to understand, or we wanted to see some motor deficits so we could actually measure how severe the insult was. And you can see here that on our sham injury, there's a little bit of a mark in the brain right after we perform the craniotomy, but you can see that there's not a whole lot of changes that happen in the brain and really not long term. But when we look at our mild TBI model, you can see that we have bruising on the brain, and that actually stays there. And we actually can still see the remnants of that impact all the way to 30 days post-injury. So you really have that on board. What's also really important to point out about this is that there is no cavitation within this model. And so that's really important to us because if you have a mild injury, as a human, you're not going to lose part of your brain. So you're not going to have a hole in your brain. That would be a much more severe model. However, mice can lose part of their brain and still look like they have a mild injury and still function pretty normally as a mouse. So we wanted to make sure that we didn't have that cavitation there, so we could understand really what's happening here. So first I wanted to interrogate our motor deficits. Like I said, I wanted to see the severity of the injury and see how it actually affected how our mice interact. So when I actually show these mice to my undergraduate students, they can't tell the difference between a sham or an injured mouse. So they function pretty normally as a mouse. They don't really look like they have much difference. So we wanted to use our beam walk to interrogate some of those more subtle differences that they have. We can use different types of beams here. So we actually have 6 types of beams. We range from small, medium to large, and then we have our square shaped bars, and then we have our circular shaped bars. And so our cylindrical really on there. So we can increase the difficulty that's there so that we can see those differences all the way down to our mild injury model. Now we can look at a few parameters. So we can look at how long it takes our mouse to go ahead and run across that bar. And so that would be our time to cross. You can see on our y-axis here. On our x-axis, you can see our days post-injury. And you'll see I only go out to 14 days here. 30 days looks much the same as 14 days. So we're not picturing it here, but we do have that data for you if you're interested. You can see that our TBI, or injured mice, featured in red, have an increased time to cross at 2 days post-injury. And so that resolves relatively quickly. It resolves by 6 days post-injury, and it's a relatively minor defect. So you could see it's a difference between like 6 seconds and 15 seconds. These mice aren't falling off the bar. They're not timing out. They're not refusing to complete the task, but they do have a little bit more time struggling across that bar. So it takes them a little longer. Another parameter we can look at is going to be our total foot slips. Now, a foot slip is considered when the mouse completely misses the bar with their foot as they're running across. And so that is just a little bit of a placement issue there. Again, they're not falling off. They're not having too many problems, but they are missing the bar when they're trying to place their foot on the bar. You can see that we have some significant increases in those foot slips from basically 0 to about 1 or 2 foot slips on average there. So we do have some variation within our injured model or injured animals as far as how many foot slips they have, but it's a minor deficit and it resolves again by 6 days post-injury. So that was important to me because I really wanted to make sure that that it's not going to proceed longer. So that would indicate maybe more of a moderate injury, something that's a little bit more severe than what I wanted to study. So we also did look at cognitive deficits that were here. And so we looked at Barnes maze deficits on here. And so I'm just going to feature Barnes maze that we did at 30 days post-injury. It's a spatial learning task there. And so I have some other data if you guys are interested in other cognitive deficits that we looked at. So in Barnes maze, we have an escape hole that's right here. And so for 4 days, we teach the animal how to navigate this maze and how to get into his little escape hole so he could go back home and hang out with his friends. The process of learning this maze, they actually do different types of patterns of exploratory behavior here. But the main goal is that the animals actually understand that there's some markers around the room, and you can see this one featured in blue here. And so they'll use that to orientate themselves on the maze because they can't use scent and they can't use tactical feel because we actually change the position of the maze and we wipe it down with alcohol every time to make sure that those type of indicators are not present for the mice. So on our day 5, though, after we have trained the animal, we take away that escape hole and then we ask the animal where does it want to hang out. And we time it for 90 seconds and we look at which quadrants it hangs out in. So I have some tracings here for you to look at. Now, our escape hole is featured right here. And so this would be our gold zone quadrant, and this would be our opposite zone quadrant. And that's what I'm really going to talk about here. You can see that our sham animals are centering their exploratory behavior during that probe trial over the escape hole. And so they know where it is. They go over there. You can see that there's some tracking that happens that they might go and investigate some other areas, but it's really centered on the same area that they know how to solve the maze. When you look at our TBI animals, they're looking more at that exploratory behavior. They're investigating the outside of the maze, so they might not be using those spatial cues as much. And this is important because there's a few different structures of your brain that are involved in this test. You have to be able to see the actual cues around the room. You have to be able to process that information. And of course, your hippocampus has to be intact as well for processing to understand where you are. Your hippocampus is like a GPS for you. So to understand where you are in space, you need to have that intact and processing effectively. When we look at our actual graphing of the time spent in each zone, you can see that our TBI animals actually have decreased time in our goal zone and increased time in our opposite zone. So definitely showing some deficits here in our spatial learning test. So next, I really wanted to understand, okay, I understand that there's some behavioral deficits that we're seeing here, but I want to know what type of immune response we're seeing, what type of cells there are, and what activation markers there might be going on there to kind of indicate what type of signaling mechanisms might be used, what type of mechanisms we could further investigate later on. So we use flow cytometry. So flow cytometry is a great tool in order to identify cells, in order to look at markers on those cells. So what we did is we isolated the brain, we divided them in half. So the ipsilateral side, or the side we injured, so that right side that I showed you earlier, or our contralateral side, or our uninjured side. And then we took it down to a single cell suspension. And that you can see the final phase of getting it down to a single cell suspension at that, so no double cells or anything like that. And then we look at our markers. And so for us, we can use CD45 and CD11B to identify where our populations are. For this gating technique, I was identifying our microglia and neutrophil populations here. And so you can see there's CD45 positive, our CD45 high expressing cells, and then CD11B positive as well. We take it over here to look at our Ly6G population. Neutrophils are Ly6G positive, and and macrophages are Ly6G negative. Now we actually stain these cells with antibodies so that they identify these markers on our cells for us. So after that, we can look at our macrophage population and look at some pro-inflammatory markers like MHC2 or CD86. And then we can also look at anti-inflammatory markers like CD206 and see how that response might change, or see how the phenotype of our cells is going to change over time. So I'm going to highlight our macrophage and neutrophil populations for this study. When we looked at our response, we saw a pretty standard response for any injury in your body. Our neutrophils come in early and they elevate quickly. And then our macrophage comes over and they start taking over that response and start coordinating it with our neutrophil population. So that's pretty standard as far as what we would expect in that acute population. That resolves pretty quickly around 7 days post-injury. And then at 15 days and 30 days, we see those cell numbers are the same for both our sham injured and our injured mice. When we look at our activation markers, you can see that we have that pro-inflammatory response elevated at our 3-day post-injury. And that makes sense. We have an insult that happens to the brain. We need to have a pro-inflammatory response. However, what gets interesting is when we start looking at our CD86 population. Again, it's a pro-inflammatory marker. And our macrophages are actually at levels similar to our sham. So no increase in the number of macrophages that you're seeing in those brains, but you are seeing an increase in the expression level of CD86. So not necessarily that CD86 is going to be responsible for the pro-inflammatory response, or that it's going to coordinate it, but that it's a signpost for us, that there is something going on within our macrophage population. They are expressing different markers and they are pro-inflammatory at that point. Apologies, I'm not used to wearing a mic. All right, so very interesting for us. And so we wanted to go further and we wanted to look at what messages are these cells actually sending to one another. And so all these changes are really happening on the ipsilateral side of the brain, not seeing it on the contralateral side. And so we wanted to see, okay, are there changes in the messaging system, and is it also isolated to that side of injury? So just as a quick reminder for people, when we actually transcribe to proteins, we go from DNA, we go to RNA, and then we go to protein. What we went ahead and measured here was RNA. And so we're going to look at the RNA of these messenger signals that these cells are sending back and forth. And we used RT-PCR. It's a relatively simple and quick method that we can use in order to look at many different signals within our population and use our samples effectively. We also have trained a high school student in order to do this, so a relatively simple method that many people in our lab can actually assist with. So it's a great method to use and a great training method for our trainees in our lab. We're just going to put that over there. All right, so when we look at our 3-day injury model, you can see that we have some elevations that are happening in our red population, our injured population. And you can see that we have increases in our pro-inflammatory response. That makes a lot of sense. Again, we have an injury to the brain. We're going to have a pro-inflammatory response that happens as our cells are coming in at that point. So acutely pro-inflammatory. However, you also see some changes in our anti-inflammatory response, which is over here. Now, that is essential to limiting the amount of damage to your brain. If inflammation actually goes unchecked and is completely pro-inflammatory with no anti-inflammatory signals, it's going to spread that damage and be really detrimental and ultimately fatal. And so really important to have that anti-inflammatory response kick in and limit the damage there. Then at our 15-day injury model, that chronic injury where we see our CD86 response that's elevated, we actually see our pro-inflammatory response change. And so you'll see that we have some other elevations. And what got us really interested is that messenger signals like CXCL10 are actually produced by our adaptive immune response, not our macrophages and our neutrophils, which are the areas that actually had some interaction with our injuries and had some increases that were happening early on in our injury model and had some activation profiles. We didn't see changes in our adaptive immune response. So that's really important to see these messenger signals because we're looking at an entire half of the brain, we are actually missing where these cells are coming in, or they're not actually increasing in number, they're just increasing in their activation level and they're increasing their activity level. And so that's really important for us to understand, especially as we look at mechanisms. And so that's where our lab is going to go eventually, is going to go into those mechanistic studies to understand how these things happen and maybe how we can treat these injuries and reduce risk. Now, everything that I've shown you is going to be kind of a global response isolated to each half of the brain, but we don't actually understand where it is, right? So we want to understand, do we have damage to our hippocampus? Are we having an immune response in our midbrain? Are we having our reward pathway affected? What is actually going on here? So we want to localize that response. We used immunohistochemistry. And so we slice the brain, we stain them with antibodies again, and they're fluorescent antibodies. So we'll actually be able to see some pretty pictures that actually Dr. Barr has provided for us over there. And so what we're going to focus on for this presentation will be the hippocampus and the ventral midbrain. When we look at our hippocampus, the area that I'll be showing you, so here's where we're going to see it on this section. So the area that we're going to highlight for you up here. And then for our actual hippocampus here, we're going to focus on our dentate gyrus and our CA1 through 3 areas. Function between these areas is really going to be essential for knowing where you are in space and understanding those spatial cues. So really important for our Barnes-Maze test, right? So when we look at those, we see increased immunoactivity in our IBA1 positive cells and GFAP positive cells. Now, what that means is we're going to see increased color on our actual stain sections in there, and we're going to be looking at our IBA1 positive cells or macrophages and microglia, and so that's going to be in green. And then you're going to see GFAP positive cells or our astrocyte population, which can actually communicate with our inflammatory response and coordinate some of those responses that are there. So when you look at our sham here, you can see what our control animals look like. When we look at our acute injury, you can see that we have increase in that green staining. Now that makes a lot of sense. We have increases in our populations that were happening there within our macrophages, right? So you're going to get increased staining, but this is at a site that's that's far away from, well, is deep to our cortex. And I want to say far away because it's a little bit close. But so here's our cortex where we're actually injuring, and our hippocampus is going to be underneath that. At our 30-day post-injury, you can see those astrocytes coming in. Now, astrocytes can participate in scar formation, they can proliferate, and they can actually communicate with the immune response. So maybe they have a role that's happening here that we don't quite know about quite yet. And so really interesting that we have that happening in our population. Now, when we look at our new and positive staining, our neuronal cell bodies, that's where it gets a little bit hard to see on these pictures because they're stained in pink. And of course, our DAPI, who also stains our nuclei, is going to overlap those areas. So I went ahead and put them in black and white so you could see a little bit better what our new and staining looks like. So in our sham animals, you can see a nice pattern formation that happens here in our hippocampus. When you look at our 3-day post-injury, and you look at our 30-day post-injury, you're not seeing those patterns happening anymore. So you're seeing a little bit of disorganization that's happening there. So what does this mean for processing within these areas? We definitely want to do some more studies on this and looking at how those cells communicate with one another, because that could really be telling us why we're seeing those deficits. Now, our ventral midbrain, and just bring this up for you again, our ventral midbrain is going to be down here, so pretty deep structure to you. And again, we're entering up here on our cortex. And so that's going to have our areas of like our ventral tegmental area within it and our nucleus accumbens, but we're just going to call it our ventral midbrain as far as our slides, just because we don't have quite as many of our anchoring points to be too specific on where our VTA necessarily is versus some of our other areas that we could be looking at here. So when we look at that ventral midbrain, again, we see increases in our IBA1 positive cells, our macrophages and microglia population. And so that could be very interesting for the development of substance use disorder. So in our sham injured mice, again, you're going to see down here, you see that increase in that green color that happens. Really obvious at 3 days post-injury and then still pretty present at our 30-day post-injury. And so really this is going to be kind of site specific. We did look at other sites that we're not necessarily involved, so we do have that staining done, and it's in our publication as well. That brings us to our research study number 2. Now, again, I told you that those areas are important for the development of substance use disorder. And so now we want to understand, are there changes to drug-seeking behavior when we look at our rodent assays? So our study is entitled Mild TBI Increases Preference to and Consumption of Opiates in Mice and is associated with a chronic pro-inflammatory neuroimmune response. Now we started out with a pretty simple hypothesis. Mild TBI increases consumption of and sensitivity to opiates. So we're going to really look at those behavioral assays here. Now, IV self-administration is a great one to look at consumption because the animals actually dose themselves with drug. And so they really control how much intake they have there. They have a lot of autonomy over that. So here is our timeline. And so when our animals arrive, they have to be trained in order to operate our operant chambers. So that looks like this. We have a little rat, and he's actually dosing himself with cocaine in this picture. You can see that he has his little hands on that lever, and a light comes on when he presses a lever. When he is training, he gets a little sugar reward. And so they understand that pressing that gives them something in return. At 5 days before injury, we do catheter implantation. We do this first because we can use NSAIDs and to control our inflammatory response and make our animals a little bit more comfortable, but we don't want that interfering with our inflammatory response to mild TBI. So by 5 days post-injury, they are no longer being treated with those drugs. So then we do our CCI TBI injury, again, same way that we did it in our previous study, and we allow the animals to heal for 5 days after that, and then they start IV administration. Now it's called short access because they have 2 hours to dose as much opiate as they like, up to, we have a safety limit there so they don't OD, but other than that, they are allowed to dose however much they would like. They are monitored completely for health defects during this time. And then at 15 days post-injury, they get sacrificed, and we're going to look at flow cytometry results again in this study. When we look at our total morphine intake over this time frame, now our morphine intake was standardized to the weight of the mouse, so that we're not looking at like just bigger mice taking more morphine. And then we looked at day of self-administration. So day 1 is actually day 5 post-injury, but it's day 1 of self-administration. You can see that they start out dosing pretty similar amounts of morphine, but then at day 2, day 3, and day 7, they have those significant increases. And then they do have a higher amount over the entire time frame. We do see some variability at the end. That actually occurs because We have some of our sham mice who have decided that they don't really like morphine, but we have some of our injured mice who have decided they really like morphine and they're just accelerating their doses. So we have some high dosing mice that are happening there as well and starting to reach our safety limits. So very interesting results that we have at our long-term time or later time point. When we look at our weights during self-administration, we have a group that we administered only saline to be able to control for the morphine intake and understand if you're injured and you're getting morphine as opposed to saline in your IV, how does that change some of your health-related consequences. And so you can see here that our injured mice who are receiving saline actually have higher weights pretty much throughout until you get to that later time point than either a sham injured or injured animals that are receiving morphine. And you can see some significant differences between our injured group that's receiving saline and injured group that's receiving morphine, both at day 3 and day 8 post-injury. And so you can see some of these deleterious health effects that you actually see in our clinical populations. Now, again, we're going to look at our flow cytometry results, and I'm going to show you again our ipsilateral site of injury pretty much exclusively in this talk, except for one study. I'm going to show you the ipsilateral site of injury. So we look at our macrophage, our neutrophil, and our natural killer cell populations. So I'm going to bring in the natural killer cells as well because we start to see some changes in another innate immune cell that's happening there too. At this point in our previous model, I showed you that there wasn't really differences between our sham and our injured mice in number of cells that were on that side. However, when we add morphine, at our 15-day or one of our chronic time points, you're actually seeing an increase in macrophages, an increase in neutrophils, and an increase in that natural killer cell population. And so something looks like it's happening uniquely in these animals. So very, very interesting results, but we wanted to understand a little bit more about drug-seeking behavior because it's not just consumption, it's how animals actually relate to the feel of the substance, relate to that dose of the substance. So we used a condition place preference model. So in this model, mice are injured in our same way. Then we go to our baseline preference. Now, mice are put in this apparatus right here. So on one side, we dose mice with a drug that we're interested in understanding whether they prefer or avoid that substance. And so we put it on our paired side. So the animal is isolated to this area on training day so that they associate that area with the response to the drug. On our unpaired side, you can pair it either with a separate substance, but we paired it with a relatively inert substance of saline in order to see how saline compared to our morphine administration. This area in the center can be closed or open depending on our use for that day. During training sessions, it's closed. During our preference sessions, it's open. So during our baseline preference, we actually want to make sure that our groups, so animals receiving different doses of morphine, actually have no preference to either side as a group average. And so we do that baseline preference model. And then animals get either saline or morphine for 4 days, get a week 1 preference trial, and then get either saline or morphine for 4 more days and get a week 2 preference trial. So just for your own information, a normal animal will establish preference to morphine at 2 weeks post-injury, not at 1 week post-injury. And there's our little mouse hanging out in his drug paired side. All right, so this is a very, very complicated graph, and I'm going to highlight just a couple of things on it. We, on our y-axis, you're going to have a change in time. And so we're going to look at how much time they spend on our drug paired side minus the unpaired side. So a positive number means they're spending more time on the drug paired side. A negative number would mean they're spending more time on the unpaired side. So significant changes between our baseline or pretest are going to actually be an establishment of condition place preference. And so you can see that our sham animals and our TBI animals actually establish preference at a lot of different doses. So on our bottom x-axis, you can see we have our sub-Q morphine doses. So we use doses. 5 mg per kg is a clinical dose. And so that's going to be clinically relevant for pain relief in our animals. And then we go all the way down to 1/10 of that dose. and see if the animals actually prefer that side. So you can see that our animals pretty much establish place preference across groups, except for one little exception over here. At 0.5 or 1/10 of the dose, our sham animals do not establish place preference at our 1 week time point. And so it looks like our injured animals are establishing a place preference earlier. Now, that might not seem super significant to you, but they've only had 2 experiences with morphine at the 1 week time point. So very interesting. When we look at comparing how much time they spent on the drug paired side at our week 1 and week 2 preference trials, now we're just going to look at sham versus TBI at those time points. And we're still going to be looking at our change in drug and time on morphine paired versus unpaired side. You see that we have significantly more time spent on our 1/10 of the dose at our week 1 preference trial and significantly more time spent on the drug side at our week 2 preference trial at both 0.5 and 1 mg per kg. So at those lower doses, we see that significant increase. So again, we wanted to understand what was happening in our immune response. And so when we look at our innate immune response on our lateral side, you're going to see an increase in macrophages that happens at 0.5 mg per kg. You're going to see an increase in neutrophils that happens at 1 and 2.5, and NK cells at 1 mg per kg. Now, you can see that there's a lot of variation here. You need to just remember that we are using low dose morphine, and so there could be changes that are happening there. There could be changes that are happening because morphine effects are dose dependent. And so we do see a curve that will happen there as far as what dose you're giving. And we also do have some evidence that there might be a difference that's happening in the brain as opposed to other organ systems with your immune response and its reaction to morphine. So you might get some of those organ specific changes. But when we go over to our adaptive immune response, which I haven't shown you any information on because we haven't seen any differences in our other models, you actually see some changes that are happening here. So in our total lymphocyte population, on our ipsilateral side, so on our injured side, you are seeing increases in our lymphocyte population. You're seeing increases from 1 to 5 mg per kg in our CD3 positive population. And that's mainly our CD4 T helper cell population. So this is really interesting. However, you'll notice that in our CD3 and our CD4 population, we actually have a pretty decreased amount of cells that's happening there. So you can see that we actually have a break in our y-axis to actually understand or to actually show all of our results. And so you actually have a decrease here that's happening across the board to our doses. And so what's the functional impact of these 2 graphs actually can be debatable at this point. So we need to understand the function of those cells. But for our total lymphocyte population, you'll see that they all have similar numbers, and I didn't have to do that break. However, with these, our total lymphocyte population has a lot of other cells that are a part of that. And so there is a possibility that they're influencing some of those total values that you're seeing as well. So a little bit more cell diversity within this result population. So that brings us to research study number 3. So that was all good. I understood, okay, we have some changes that's happening in the immune response, but it's only giving me a picture at 15 days post-injury. I'm not seeing how that changes in the acute response phase. I'm not seeing how this changes over time. And really, within our clinical population, most individuals, or many individuals who develop a substance use disorder actually are prescribed opiates. And so they're going to be getting that clinical dose. They're not going to be accelerating their dose really in those early days. They're not going to be receiving low dose morphine at that time point. So I wanted to design a different study that I can understand how that's happening. So our research study number 3 is entitled Chronic Dose Morphine Increases the Pro-Inflammatory Response After Mild Traumatic Brain Injury. Our hypothesis was that there is an increase in macrophage and neutrophil recruitment and the inflammatory response to injury in mice that receive a mild TBI with chronic morphine exposure. So here's our timeline. Our animals arrive and then they get trained on injection training. The way that I give morphine is actually a subcutaneous injection. Now, animals don't like restraint. They get very stressed out during restraint. And so we actually train them with an inert substance before we actually inject anything else so that they can kind of settle down and not have such a stressful response. And so that's not going to be a confounding factor as much for these results. We then go ahead and induce injury, and we look at our immune cell phenotyping, and we continue that out until day 15. So our chronic time point again. Our mice immediately after injury are going to receive 5 mg/kg, our clinically relevant sub-Q dose. They're going to receive that twice daily. That's because we want that morphine to be active and to be on board throughout the day and not to have those highs and lows of withdrawals. Because like I told you, the other animals are intermittently dosing. So 2 hours a day, or they're getting 1 dose of morphine and then 1 dose of saline. And so they actually have different levels of morphine in their system at all these time points. So we wanted to kind of eliminate that withdrawal that happens there, because that could really affect what's happening with your immune response. We also did behavior testing. I'm not going to focus on that here, but if you'd like to see it, we do have some interesting anxiety responses and such that were happening in that population. So flow cytometry, our old friend, is going to be used here. You're going to see a lot of different symbols that are used on these graphs. I have what they mean over here. They are going to refer to the morphine exposed injured mice against our other groups. Now, I'm not going to go into too much of what those symbols exactly mean on this one, and the interpretation. That's more for the paper if you want to go to that one. But so we're just going to focus on that we have increases within that population and where they're significant. So when we look at our innate immune response, our macrophages have more cells within them at the acute time point at 3 days post-injury. But we actually see a unique response that happens at our 15-day time point. And so you see that elevation of macrophages that happens again once we have that decrease at 7 days. And so very interesting for us, what's this macrophage population doing? What's its functionality? In our neutrophil population, you can see that we have that elevation here. However, our TBI with saline is also elevated here. You can see TBI with saline is now in white, where our TBI with morphine is in red. And you can see in our NK cell population, we have an increase that happens at our later time point. So interesting dynamic responses that are unique within our injured morphine exposed population. So we wanted to look at our activation markers again, right? We want to understand what's happening within that population. Now, we have some typical responses that we were expecting. I didn't show you Ly6C before. We have elevations in our Ly6C marker activation on our macrophage populations at the acute time point in our TBI versus sham injured. So we see that again, that happens here. We just have a bit more of an elevation that happens within our TBI morphine exposed group. But what's interesting is that our CD86 expression actually happens here at our acute time point and not at our long-term time point. So different type of functionality that's happening again. When we look at our MHC2 population, we actually see that elevation happening at 3 days, which we expected. But then again, we see an activation time point at 15 days. Now, that doesn't mean that MHC2 is again directing that response, but it is signpost saying that macrophages are doing something and we need to understand more about their functionality. So really showing us that there's some type of pro-inflammatory response that might be happening there. When we look at our adaptive immune response, again, we see some interesting results. We see an increase in our total CD3 positive T cells on the ipsilateral side. Now, when we look at these results at 7 days, regardless of whether you are injured or whether you are a sham animal, we have an increase in that population that happens. It's transient. It goes away at 15 days. I've seen that in our study number 1 before, and it wasn't unique to any different treatment protocol. However, when we go to 15 days post-injury, that elevation is maintained in our TBI morphine exposed mice. So maybe we're getting some more of that adaptive immune response contributing as well. But what was interesting is that as opposed to the results I showed you earlier, it looks to be dominated by the CD8 cytotoxic T cell population, not our CD4 T helper cells. And so a different branch of the adaptive immune system is being involved there. And so what might be the functional changes that we're seeing at that point too. So seeing a little bit different time point there. There are some ideas that we've thrown around as far as why that might be. And really we could point to some of that withdrawal and elevation time points that we saw in our CPP and our self-administration protocols, where this is really a constant exposure to morphine. And so maybe some differences in how we actually dose the animals. When we look at our cytokine response, again, we're going to look at our RT-PCR, our messenger RNA system. And so we're looking at changes that are happening. We relate them all to our sham injured mice. And so we can understand how we see fold changes within our TBI morphine and our TBI saline population. And so you can see increases and decreases that happen within these populations, and we compared them to each other. At our early time point, you can see that we have some pro-inflammatory responses that are happening. You see our TNF alpha population that's significantly increased, and our CCR2, CCL2 population is actually coming on board. And it was on board with our other one as well, which makes sense because CCR2, CCL2 population is actually a mechanism to recruit macrophages into the brain. Makes sense. We have increased macrophages. We're going to have increase in the population that's recruiting them to the brain. Now, when we go to our 15-day time point, you'll see that we have a significantly different set of messengers that's happening here. And so understanding what the functional changes are and what is actually happening with that is going to be really interesting. You can see we have anti-inflammatory and pro-inflammatory changes that are happening here. And it's really interesting because morphine typically is immunosuppressive. And so we expected to see an increase in that in our anti-inflammatory population. However, we see some variations that are happening at that long-term time point when you have TBI on board. So really, really interesting. Really want to explore that further. Now, our next study, I've been talking about that we want to look at function this entire time. So I finally got to look at function. And we love our macrophage population. They're heroes in our lab, and we adore them. So we're going to look at our macrophage phagocytosis. Macrophage literally translates to big eater. And so phagocytosis just refers to how they engulf things. When we look at our macrophage, the way that I evaluated this is I have these beads. They're strep-avid encoded beads. Macrophages will eat them. They are identified with a fluorescent marker. So when I look at them by flow cytometry, I can identify beads within that population. Now I put another marker on top of them so that I can identify where the beads are within this population. So when a macrophage eats one of my beads that are double labeled, now the macrophage is double labeled for both the bead marker and that secondary marker. However, if the macrophage does not eat the bead, they're still double labeled, right? So we have to add a quencher. So we add a quencher, takes away our secondary signal, and now I can tell if the bead is on the outside of the cell and not engulfed. That's really important to understanding the functional changes that are happening within this population, because as we found out in some of our studies, beads like to coat pretty much every cell. So, all right. Now, when we looked at our macrophage phagocytosis, we isolated our white blood cell population, much like we did in our flow cytometry. But instead of going straight to staining, we took that population and incubated them with the beads. And so we made sure that we were using a similar population size, both for all of these different groups that you're going to see here. So again, we're going to have a number of cells on our y-axis, and then our groups are going to be on our x-axis. Now in our macrophage population, you can actually see that we have a decrease in phagocytosis. Now, I was not exactly expecting this. I was wondering if there was going to be an increase in phagocytic activity, maybe some neuronal remodeling that was happening. We did this at 15 days post-injury, so I was hoping to see some physical changes that were happening. But what I saw was actually a change in completely the opposite of what I thought was going to happen. So very interesting for us, but that means that the functional changes might not be in this population. Or they could actually be eating specific items. And so maybe when they have been in an injured animal exposed to morphine, they just don't like to eat strep-avid encoded beads. Maybe they want to eat something else. However, when we looked at our microglia population, I was also surprised because we don't see phenotypic changes within our microglia population. That's why I haven't shown you the data on our microglia population. It's not very interesting, no significant changes. However, when you look at functionality, you actually see this decrease as well. And so really was showing us that there might be some mechanisms at play here that flow cytometry just wasn't exposing to us on those studies. So very, very interesting results. But again, we have to look at some more parameters to really understand what this means and look at some other functional assays for our macrophages. Also, this was done was done ex vivo. And so when you take inflammatory cells out of a brain and you actually culture them, you've changed some of what's going on there. And so you can't actually activate those cells just by removing them from their system. And so definitely need to understand what they're doing inside of the animal as well as ex vivo. So really need to contextualize these results. Now we looked at blood-brain barrier integrity as well at this later time point. And so I told you before, when you have an injury to the brain, you can have compromise of that blood-brain barrier. Blood-brain barrier is very important to keeping nutrients going into your brain and keeping toxins going out of your brain. And if there's damage there, you're going to have a different efflux and influx of those substances. So what we did was I used an Evans blue stain. I went ahead and injected it in the tail vein right before I sacrificed the animals. If there is an issue with blood-brain barrier integrity, you can actually, the brain itself will turn blue and you can measure that. So I measured fluorescent intensity of the actual stain within different blood or within different brain areas. And so you can see here, I looked at the hippocampus because that's been involved in some of our deficits we've seen. I went to look at the midbrain area because I wanted to see if there was something happening within those structures, those deeper structures. And I also looked at the cortex. Now, you can see that we don't really have significant changes that are happening within our cortex or our hippocampus at this longer term time point. However, we do have that significant increase in Evans blue staining within our midbrain area. And so that really kind of suggested to us that there was something going on here and there might be some compromise of that blood-brain barrier. However, we want to do some more tests. So we want to understand what's the size limit of those cells that can actually cross the blood-brain barrier. And then we can start looking into some other factors that happen there. We can look into the cells that actually comprise the blood-brain barrier, like astrocytes, pericytes, things like that. So we can look at those cells as well. And so that really gives us a future direction to start examining too. Now, I've shown you the studies, so let's talk about exactly what those studies mean. When we look at our conclusions, the contributions of the present data are significant because they provide evidence to link the inflammatory response to increased susceptibility of substance use disorder with opiates. And so in our first model, I developed a model of mild TBI that has chronic inflammation that correlates with long-term cognitive deficits. Now, this is important not only for substance use disorder, but we can actually apply it to other studies within our lab. We like to understand how Alzheimer's happens. We want to understand how different brain proteins actually contribute to these increased risk of these neurocognitive deficits that we see in our long-term in our patients. And so we can use this model for more than just substance use disorder. So that was really important for our lab. In our second study, we demonstrated an increased consumption of sensitivity to opiates after mild TBI. And so really what that study does is that we can, once we identify treatments or we want to modulate the inflammatory system, we can see how that affects our drug-seeking behavior. And in our final study, we looked at, we modeled changes in the pro-inflammatory response to TBI with the administration of morphine from the acute to chronic stages. Now that really can help in identifying where we should interfere with that response. So where we can modulate that, where we can use treatments, and then we can also look at mechanisms. So we can use that to investigate what's happening with the functionality of these cells. So we can really be specific about how we're treating this injury. So what I basically did is develop a working model for the lab. So we have our inciting incident of mild TBI, incites a pro-inflammatory macrophage response. That leads to increased sensitivity and consumption of opiates. And then that in turn alters the immune response. So we've seen that, right? What we don't understand is how, or the mechanism behind these changes in our inflammatory response and the increased consumption of opiates. And this really is just a cycle. They feed into each other. It just becomes a circle that becomes worse. And so we want to understand the mechanism between these two so we can interfere there and decrease that risk for at-risk populations. What we think is happening is accelerated neuronal remodeling. So actually changing how our neurons are interacting with each other and actually processing information and signaling to one another. So I want to say thank you to my contributors, but I also want to highlight some of the things that they've done for me. And so some of you know this, but my first lab that I was in actually ended in a little bit of heartbreak for me. And I called my parents because, of course, they give me the best advice. And they told me to go to people I trusted. And so I did. I went to my head of department, Dr. Rutherford, and he really helped me to tell me that this happens to other people. It's not just me. And so that made me feel a lot better. They helped navigate that transition. I went to Dr. Mizell, who's on my committee, and he really helped me understand that like it was okay to change and he was still going to be there for me after I changed to a new project. And then I went to Dr. Molitor, of course, and he told me he had a P&I training grant that would be perfect for me. And so introduced me to some of the people who worked on there. So I went and talked to Dr. Charon, and he told you I trained in his lab before, but what he didn't tell you was that while I was training in his lab, he told me just to stay in his lab and run all my experiments in there. So by the time I interviewed with him, he told Dr. Molitor that he just wanted me in his lab, period. So he definitely wanted me as a student. And then, of course, I trained with Venkat, who's been there with me every step of the way, and I can't thank enough for everything he's done. I got Dr. Lowe as my other advisor, and I swear one conversation with him will have you just seeing new possibilities for the future, open up different avenues of research, and he really made me feel valued and respected, as did Dr. Andrew Crane, who was a postdoc in the lab at that time. I also met Dr. Grand through that, and he really valued my opinion on multiple projects. So we worked on many things like stroke models with Dr. Var. And so really helped me to see that my contributions were valid, and we really worked together a lot. So opened up both of our avenues for research. Dr. Brown, of course, I swear you are just the kindest person in the world. And he teaches pharmacology for us. So he's been there for me since I've had so many questions for how to keep it on board all the time, how to understand the effects that are going on. And of course, Suzanne was there to relate to and commiserate with, because she just did her PhD as well. So I could call her when I was frustrated trying to write a thesis. So, and to all the undergraduates, graduate students who helped me along the way, and my friends at vet school and grad school who made a home here in Minnesota for me, thank you. Couldn't have done it without you. Sorry, I'm getting emotional now. Of course, I have to acknowledge my wonderful husband, who's been supporting me through this. Of course, he's pointing to himself right now as well. And I always talk about my constant companion, Sly, who has gotten just so good at data analysis. I really couldn't have done it without him. He actually wrote part of my thesis for me. So really great companion. And of course, our newest recruit, Poe. He can't focus. He doesn't really help on anything, but we have a lot of hope for progress. So he has a lot of stuff going for him though. So I will take questions while my husband prepares my broadsword for me. So. And I actually do have a sword, so, you know. Yeah. Thank you. As far as what shows the most promise, modulation of the immune response definitely does. And we've seen that in ethanol, and we've seen that in cocaine models. And so I really do want to target those cell populations. And since there's increases at different points, we could actually target different ones. Because we have macrophages involved in so much of the work, we do want to target that population, but we could also target the T-cell population. We have some stem cell work that goes on in our lab that modulates immune response. And so that provides some some avenues for us to go down. I'm sorry, what was your first question? Oh, yeah. So really with those high risk populations. And so Walt and I had talked before about having soldiers who are deployed to combat. Even hearing a explosion, you are close enough to get a mild TBI injury for that. We've demonstrated in our animal models. And so understanding that and using preventative measures and populations we know are getting those type of things, or populations that are involved in sports that have high contact. Kids who play football, kids who play hockey, things like that. We do see that once they get to the college level, that's actually inappropriate control if you look at them before and after their season, because they already have damage at the preseason when they're coming into their college careers. And so possibly going back even to high school when they actually start those. And understanding what's happening there, maybe doing some intervention there. Especially, we see some really big differences in kids who are injured under 13. And so that's a really big at-risk population. Yes. Mm-hmm.
But then you also have kind of, with conditions-based practice, kind of the opposite now, patient pastors having more sensitivity for the response, and if the administration, if there's supplementary. And so I'm kind of like playing in my head with whether or not this is a compensatory thing where now you have the residents can't escape, or whether it's they want to knock down the immune response, so they're taking kind of bring that down. So all that comes down inflammatory responses.
Yeah, so we do want to target more of those areas. And so we definitely want to understand what's happening there. And if those deficits that we see, or those changes that we see really in the hippocampus is consistent across there. And maybe if it's that, the rewarding system is just that powerful that it's, overcoming some of those deficits that we saw. And our animals there, they didn't show a complete lack of understanding where they were in our Barnes maze, because you saw that they were spending more time in their goal zone than in their opposite zone. So maybe there is a bit of function that's happening. It just might not be as robust as in our sham animals. And so really understanding those targeting areas and how important those areas are, what the functional changes in them are going to be, and then altering those functional systems. So can we target those areas? Can we stimulate those areas? And then can we look at some of those responses? And so that's really where a lot of our group wants to go, or using behavioral tasks that are more specific to different areas of the brain. And so I've met with a lot of experts really in this building to try to understand that and try to target those areas. But we really need to identify target areas and identify behavioral changes before we can move on to specifying what we're going to do. And so that was really what my project was doing, was like building it up so that somebody could take it over and really start identifying and just understanding those mechanisms that are there and understanding what's really important to that. So yeah, so we want to go there. I've done enough. My committee said they didn't want to give me any more work, and I think that's pretty much when you get your PhD, right? So yeah. Yeah, so as an aside, we used all female mice here really to fill the gap, because males are used a lot in research, and they're really used in TBI research because a lot of people associate TBI with males, right? They do high risk activities, or kind of dumb activities, so and they get injured more often, visibly more injured more often. But we've been finding that females actually have a whole other array of injuries that happen. So understanding the sex differences is going to be really important. And a lot of the ones that involve females and high populations of getting injured are going to be like our domestic violence situations. And so those are ones that we're really not even identifying very well, right? So understanding the sex differences is going to be essential kind of moving forward. I used 8-week-old mice mainly because we're targeting that kind of young population. So that adolescent soldier population, you're really looking at like teenage to mid 20s years, right? So we want it sexually mature, but I didn't want old age mice. What we do want to understand is, okay, when you go in and you have a mild TBI, you're not going to be prescribed morphine, right? Like that would be a little bit of an overstretch. But what happens when you get a mild TBI early on and then later responses? So we targeted this early on to see if we see changes that are happening and see a little bit more promise of robust results that are happening here. But another avenue that the lab wants to go into is injuring early and then waiting until those long time points, doing the drug seeking behavior again and understanding how that changes long term and if that does correlate there. Also multiple injury, because we definitely do have multiple injuries that happen sporting and being deployed to war, things like that. So we want to understand those models as well. And so those models are also being developed. Just not my job. You can ask me what the little stuffed animals are. What am I going to do after I pass? Well, fingers crossed. Max has been vaguely threatening for the last few weeks. So, fingers crossed on that one. But I'm going back to veterinary school. So, what a way to celebrate. Go to clinics. Yeah, we have a couple of vets in the room. They're looking at me like, oh, no.
Got some questions from the Zoom audience.
Yeah, no, I get a couple of weeks and then I have to go back. Yeah. Yeah. So there weren't significant differences. And I think maybe if we increase the group size, because we did an N of 9 in each group. And so we can definitely bump that up. We might see some differences that happen. It looked like there was a trend that our animals who were sham injured were actually decreasing their time to finding the escape hole, where our TBI animals start to plateau a bit. And so it looked like there was a trend there, but we'd have to increase the group size in order to understand that on that part. And I mean, you run barns maze. It's a little hard to use those larger group sizes just because there's only so many hours in the day and you can't spend that much time in a basement. So 12 hours was enough. So there is pumpkin bread with butter that's still there. There is clam dip that we made as well. There is also butter cookies that's there. And then champagne and apple juice, though I don't know. They were talking about already popping some of those bottles. So I don't know what's happening there. So, but that'll all be in the niche next door. Yeah, I know. I applied and then they were telling me that I need all these signatures to sign off and stuff. And yeah, so we got the approval. So that'll be over in the niche. Apparently we're only allowed to do 45 servings. So just, I mean, I'm not going to judge. So, whatever you guys want to do, you go for it. All right, and bye everybody on the online one. Thanks for coming. I mean, sometimes. Oh, Kent, you're asking when do they become addicted to morphine? We actually just do drug seeking behavior. Addiction is not a thing for animals. We don't use that terminology. We use drug seeking behavior on that instead for that. So it's a little bit different because rats are different from humans, right? Yeah. Sure.